Here is an overview of plant immunity. Plant pathogens include fungi, protists, bacteria, and viruses. These are a part of nature. In order to protect themselves, plants have their own form of immune system. Today we will focus on the plant's constitutive and induced responses to pathogens. The first line of defense against pathogens for a plant is its outer surfaces. The stem and the leaves of a plant are covered with cutin, suberin, and waxes that assist in keeping pathogens from getting into the inner tissues. These physical barriers are used as defenses. These are classified as constitutive. Constitutive defenses are not always enough to stop a pathogen from getting to the plant. This is when the plant uses its induced responses. Induced responses are chemical responses that are triggered by pathogenic molecules called elicitors. These are usually peptides made by bacteria or cell wall fragments directly from fungi. Plant immunity can be general or specific. Constitutive defenses fall under the general category, but induced responses may fall under the general category as well as the specific category. General immunity is triggered by elicitors, called pathogenic associated molecular patterns, or PAMPs, which lead to signaling pathways when bonded to receptors. The pathways can lead to many responses, including formation of nitric acid and reactive oxygen species, polymer deposition, hormone signaling, and changes in gene expression. With formation of nitric acid and reactive oxygen species, binding results in the production of nitric acid and hydrogen peroxide, which are toxic to some pathogens. With polymer deposition, polymers like lignin are used to block plasmodesmata, preventing the ability of pathogens to spread from cell to cell. With hormone signaling, it is exactly how it sounds. With changes in gene expression, coding results in the production of phytoalexins, which are small molecules produced by plant cells near the site of infection. They act as an antibiotic toxic to pathogens. Now for specific immunity. Specific immunity is genetically determined. The genes that code for elicitors are called aberrance genes. There are hundreds of these that vary greatly depending on the pathogen and strain. There are also hundreds of resistance genes, which are the receptors for the elicitors. The relationship between aberrance genes and resistance genes is called gene-for-gene resistance. If this receptor-elicitor bonding does not occur, the plant will not recognize or turn on its defenses. Specific immunity also often takes defensive responses a step further. Cells around the site of infection undergo apoptosis. This is called a hypersensitive response. This response is effective in that it deprives the pathogen of nutrients, but often leads necrotic lesions or areas of dead tissue on the plant. On the other side, specific immunity may lead to systemic acquired resistance, which is a general increase of resistance in the plant to a wide range of pathogens. Salicylic acid initiates this defense response. It is triggered by a receptor binding in both general and specific immunity. One type of systemic acquired resistance is the triggering production of PR proteins to stop the spread of infection. Another type is slightly more specific and only for virus whose genomes are made up of RNA. In this case, the plant will use its own enzymes to convert the viruses to RNA from RNA to small interfering RNA and then break down the mRNA of the virus to prevent its replication. That was an overview of plant immunity against pathogens.